Hello, my name is Seth Gammon, and I'm a Product Application Specialist for Carestream Molecular Imaging. The talk I'm going to present today is Introduction to Optical Imaging for Investigating Cancer Biology and Relevant Pathways. Why are we interested in doing in vivo molecular imaging? The main reason is to collect better data. The data is better than in cross-sectional studies because you're gaining a true history of the process. Typical optical molecular imaging techniques are non-destructive. Thus, you can repeatedly measure the same animal over and over again. In addition, you can couple information not only about time, but also about where in space this molecular event is happening. This gives you, as I said, more information than you could ever gain from a cross-sectional study, but also practically it gains you better statistical power. That means fewer mice or more complex questions than you could ask before. There are three very broad reasons why you might want to do in vivo imaging. The first is cell trafficking. Next, you may want to look at gene expression. Is a gene going on or off or up or down in the context of some disease state? And finally, you may want to optimize drug or nanoparticle delivery in vivo. The imaging sciences range from microscopy at the very small to astronomy at the very large. These sciences are all united by the fact that you're detecting an optical signal over a camera noise background. We're going to focus today on in vitro assays and primarily small animal imaging. With small animal imaging and in vitro assays, you're doing great science. And the reason it's great is that great science utilizes all the relevant techniques that you can bring to bear on a question, whereas good science only utilizes one technique at a time. Molecular biology will still have to be done. You'll have to continue to do Western blots, perhaps multiplex Western blots, in vitro fluorescence microscopy, as well as in vitro cell-based assays. However, molecular imaging becomes another approach to study the biological questions of interest. That includes localizing an event both in space and in time, whether it's a tracer or an activatable probe or a metabolic probe like FDG. In addition, phenotypic imaging can be done, and those phenotype data can be recorded for future analysis. So rather than just writing down in a lab notebook that a tumor has grown over time, this can be recorded through phenotypic images such as CT, SPECT, PET, and MRI. In addition, with the Kodak in vivo instrument, you can also collect this data through X-ray acquisition. And that can be done at whole animal zooms or zoomed in on just a small region. Thus, we like to think that there are two really big requirements for imaging disease models. The first is to observe and record the bulk physiology or the anatomy under study, whether by multicolor reflectance imaging or by X-ray imaging of the whole mouse or individual target sites. In addition, it's then important to detect and measure a molecular event, typically through a luminescent, fluorescent, or radioisotopic probe, that reports on some biologically relevant disease process. The Kodak in vivo multispectral FX thus is a one-source instrument for all of the critical areas of a good in vivo experiment. It's a one-source instrument for Western blots, such as chemiluminescent or multiplex fluorescent blots, one source for molecular imaging, such as radioisotopic imaging, fluorescence imaging, or luminescence imaging, one source for phenotypic imaging by X-ray, bone density, or reflectance. The instrument is illustrated here. You have a fluorescence excitation source from a xenon lamp that provides broad, stable excitation with a 29-position filter wheel, 28 of which are filled with bandpass filters. Above, you have an X-ray source Then you have your animal imaging chamber that's compatible with either injectable anesthetics for your small animals or isoflurane or other gas anesthesias through these ports. In addition, there are optically useful ports for feeding wires or catheters into the animal imaging chamber that you can then use to help further study and characterize the mouse. Below is a closed optical path imaging chamber where the liquids or other solids from the animal imaging chamber are prevented from entering into the optical path. The image system offers 5 imaging modalities: fluorescence, radioisotopic, chemiluminescence, reflectance, and high-resolution X-ray, although today we're only going to focus on 4 of the 5. There are a couple of major challenges for optical imaging in vivo as opposed to optical imaging in vitro. Number 1, absorbance is a major problem when doing in vivo optical imaging. That really isn't an issue with things like Westerns or fluorescence microscopy. In addition, scattering of light limits your spatial resolution, and autofluorescence and autoluminescence limit the ability to detect a signal over an endogenous background. Here's a graph of the absorbance properties of tissues, and as can be seen, in the blue and green, which are a lot of typical probes from in vitro microscopy work, you have very strong absorbance and scattering of common chromophores like hemoglobin, myoglobin, and melanin. As you move out to the red and the near-infrared, these absorbance and scattering properties are dropping off by log-fold changes. However, as the wavelength gets too long up into the infrared, you then have strong absorption by water. Mouse autofluorescence, or small animal autofluorescence, is also a problem. Here you can see the autofluorescence levels from a blue-green filter, a red, and a near-IR filter. As can be clearly illustrated, it would be very difficult, and indeed is difficult, to detect an optical signal above this high background in a blue-green filter set. And red is a bit better, and near-IR is the best. Thus, the near-infrared represents a sweet spot for in vivo optical imaging where you have maximal transmittance of light and minimal autofluorescence. What's exciting is that there have been several new fluorescent proteins reported in the literature that can be excited in the red and detected in the red or near-infrared. A very bright one from Roger Chen's group is DS-Red and TB-Tomato, which excites in the orange and emits in the red. MK2 and M-Plum are fluorescent proteins, M-Plum from Roger Chen's lab, MK2 from Evergen, that excite in the red and emit in the near-infrared. IFP is a new fluorescent protein recently published in Science by Roger Chen's group that excites in the near-IR and emits in the near-IR, although its brightness per molecule is slightly less than those other probes. There are 4 very broad classes of probes and experiments that you might do for in vivo optical imaging. The first we're going to talk about is utilizing genetically encoded reporters. Zhang et al. Has published a nice study demonstrating the power of labeling tumor cells with DS-Red as a genetically encoded reporter. They looked at 2 different cell lines, PC3s and DU145s, and tracked them over time. They were able to clearly and quickly observe that the PC3 cells metastasize much more quickly than the DU145s. In fact, as illustrated here, the DU145s really just create one large mass of tumor, whereas the PC3s were clearly metastasizing to different sites very early on. They then zoomed in to determine whether these metastases were in other organs or whether they're in lymph node sites. And in fact, in a zoomed-in view, they were able to see the red hotspots on lymphatic sites. They then utilized this information to guide their histology, and they were able to confirm that the cancer cells here detected as brown when looking for human antigens in a mouse lymph node were found in PC3 cells, but not with Du-145 cells. They then hypothesized that perhaps VEGF expression and excretion might help cause this metastatic phenotype of the PC3 cells. So they did Western blots in both cultured cells and xenografts for VEGF-A, C, and D. Indeed, PC3 cells overexpressed VEGF-A much more strongly than Du-145 cells. They then hypothesized that the lymphatic cells, which were known to have VEGFR2 receptors, were being activated for migration to and from the tumor site by this excretion of VEGF-A. To test this hypothesis in vitro, they did a co-culture of LECs, lymphatic cells, and PC3 cells. They then created a wound site down the middle of the culture and watched what happens. By 25 hours, the PC3 cells have migrated to the interior of the wound site, and the LEC cells have followed them and begun to fill in. However, whenever an anti-VEGFR2 blocking antibody is put in the culture at time 0, only the PC3 cells are able to enter into the wound site. The LECs aren't recruited. Thus, they were able to learn some really novel biology about how these tumor cells may be metastasizing to different sites depending on the genomics and the proteomic expression profile of the tumor cells. Another very exciting recent discovery is what I like to call the rediscovery or return of beta-galactosidase as an in vivo reporter. Many mice and cell lines have been created for studying gene expression or cell localization by conventional optical microscopy because beta-gal converts many substrates into a blue product that can be observed by optical microscopy. Obviously, this isn't of great use in a live animal. However, Josserand et al. Took advantage of DBAO galactose, which serves as a substrate for beta-gal and then becomes near-infrared fluorescent. They were able to detect really very few cells in vivo, and what this really means is that rather than recreating luciferase mice or GFP mice or new fluorescent protein mice, researchers can simply look back into the catalog of available beta-gal mice or beta-gal cell lines and immediately begin utilizing these in vivo. The next reason one might wish to study animals in the context of a live time course is to optimize or drug delivery. This group was interested in creating nanoparticles that were able to bypass a permeability barrier. MDR1 is often overexpressed on tumor cells that have been selected in standard cancer chemotherapeutics. MDR1 presents a permeability barrier to cationic lipophilic drug molecules, and unfortunately many of these normal cancer therapeutic drugs are blocked by MDR1. In order to circumvent this barrier, they created nanoparticles that incorporated paclitaxel and ceramide. They were able to demonstrate in vitro that the combination of paclitaxel and ceramide was enhanced by packaging this combination in nanoparticles. There's a big question, though, in tumor biology as to how well a nanoparticle will indeed be delivered to a tumor. Some groups have reported a phenomenon known as enhanced permeability and retention of nanoparticles, whereby the leaky vasculature of the tumor enhances the delivery of these nanoparticles to the site. However, it's been well known for many years that solid tumors also have much higher interstitial pressure than surrounding tissues. And thus there's a constant battle between the enhanced permeability and the higher interstitial pressure when trying to deliver nanoparticles. Thus, it's important to monitor in vivo in as many different tumor cell and tumor models as one can how a nanoparticle will get into a cell, into a tumor, and to what extent it gets into that tumor. This group labeled their nanoparticles with fluorescent probe and injected them in vivo. As you can see, because these nanoparticles aren't targeted, they're distributing throughout the whole animal. However, good news is that the nanoparticle is indeed being delivered to these tumors at 1 and 6 hours. It's not being excluded. Another group recently published in Nature Medicine that they wanted to create nanoparticles that would target tumors specifically, as well as deliver a payload of siRNA. Their model siRNA here was siRNAs against GFP. The iron oxide core is useful for imaging by MRI at some future time point, perhaps if it gets to the patient. However, to optimize the delivery and specificity of this probe, they put on a Psi 5.5 label to image the delivery and optimization by optical imaging. There's an MPAP peptide attached to help target it to the specific tumor cell type. The first experiment is to just see if the nanoparticles get to the tumor and are they specifically targeted to the tumor. In this case, they were able to confirm that because the nanoparticles were injected IV, they could track the fluorescence and see 2 specific hotspots with good specificity at the tumor site. This image also confirms that these 2 tumors, which will be important later, received relatively similar doses of the nanoparticles. Those solid tumors were 9L tumors, one of which expressed GFP, which should be silenced by the siRNA. The other expressed RFP, which should not be silenced. Thus, if the siRNA is specifically being delivered as well as acting specifically on the target protein, then one should see the lights turn out in the GFP cell line, but stay on and get brighter in the RFP cell line. In fact, this is exactly what you can see. Here, the fluorescence intensity of the RFP cell line gets brighter as the tumor grows, and you can see the GFP is dimming over time. Thus, in one very simple and elegant experiment, they were able to determine that the nanoparticles were selectively delivered to the tumor, as well as the fact that the siRNA was selectively targeting a specific gene and not just shutting down general gene expression. They then did some critical things for in vivo imaging, which is to confirm ex vivo at the key time point that they were in fact delivering probe and there wasn't some optical artifact, as well as confirm that the gene expression was truly silenced by some independent method. They did that by looking at the near-infrared fluorescence ex vivo, as well as checking the fluorescence of the tumor's GFP and RFP status by microscopy, and finally confirming by qRT-PCR, the gold standard for RNAi, that you had silencing. In this publication, the group wanted to determine if there was a way to get peptides across the blood-brain barrier. It had been previously reported that the peptide would cross the blood-brain barrier, but it really, in further publications, was not shown to cross the blood-brain barrier to any significant extent. Thus, they methylated the peptide and targeted it to the blood-brain barrier through this combination of a myristoyl group and peptide. They labeled the peptide with Psi 5.5 in order to optimize the delivery of the probe very rapidly. Indeed, after several iterations of concentrations and time, they're able to take a near-infrared image of light being emitted from the head. Overlay confirms that this is very much a brain-like image. The key experiment is they then took out the brain, confirmed that it was hot by near-infrared fluorescence, and then translated those brain slices over to a fluorescence microscope. Because their peptide was already fluorescently labeled, they didn't have to do any other type of staining for their probe. It directly translates to the fluorescence microscope. And this is a big advantage of fluorescence over luminescence imaging, because with fluorescence you can rapidly translate back and forth between in vivo fluorescence imaging, FACS sorting, and FACS counting, and fluorescence microscopy. Here one can see that the peptide is indeed being taken up by cells and exhibiting its classic nucleolar staining, but what's critical here is that the endothelial cells in the blood-brain barrier, stained green by an antibody to endothelin, are not in the same place. Thus, the peptide has not only gotten to the brain, but they were able to prove by fluorescence microscopy that it crossed the blood-brain barrier. They further went on to confirm that this wasn't just a dye by re-extracting their label and confirming its stability by HPLC. Another group wanted to determine if their molecular donuts, in fact, didn't target the gut. Really what they wanted to know is whether the hydrophobic toroids that they had synthesized went anywhere in the body or where they went. To do this, they took their hydrophobic toroid and labeled it with Psi 7, which is a near-infrared fluorescent label, tail vein injected it, and then observed by in vivo fluorescence imaging that it was localizing heavily to the liver and to some extent to the lungs. They were also co-localized this signal on an X-ray in order to clearly see to the untrained eye that this is the liver and this is the lungs. By looking at the autofluorescence, here coded in green, of the gut, they were able to further help co-localize their optical signal. They did all this by utilizing spectral unmixing and then overlaying it on an X-ray signal. As I mentioned before, even in the near-infrared and red, there's still autofluorescence, even though it's much lower than in the blue and green. Thus, whenever you're working in the near-infrared, you can either utilize spectral unmixing to remove autofluorescence or track multiple fluorophores at the same time, or both. If one is working in the green or yellow range, the spectral unmixing will pretty much just be utilized for removing unwanted autofluorescence. Chemistry molecular imaging utilizes excitation-based spectral unmixing in our system because there's more information typically in the excitation side of a probe than in the emission side. The first example is a trivial one, which is to deconvolute multiple fluorophores in vitro. Here, we have our Excite 761, 691s, and large Stoke shift dyes, and in Vitrogen's quantum dots, all arrayed on a 96-well plate in various dilutions. If one just looked at this image, there's no way to tell which probe is which unless I have it labeled. If multiple excitation filters are utilized to capture fluorescence images, a stack of images can be built up from 400s out to the near IR. The computer can then stack all of these images up, look down through the stack at each pixel to generate an excitation spectrum, then utilize the information both in the shape of that spectrum as well as the intensity to deconvolute out each fluorophore into its component parts. The in vivo problem is obviously much more difficult, but still approachable. Here, the spectral unmixing will be utilized to remove the skin autofluorescence, gut autofluorescence, which isn't even visible in this image, as well as split out the fluorescence intensity from the Excite 761s and 691s. Again, a stack of images is required at different excitation wavelengths, and then deconvoluted into their component parts. Here, the skin autofluorescence is coded as yellow, the gut autofluorescence is coded as green, and the Excite autofluorescence is coded in red and light blue. Because the mouse is never moving during this process, it can be directly overlaid over our X-ray or our reflectance image. And that's how this group was able to mark the gut here with green and remove the skin autofluorescence, really leaving only the fluorescence coming from the liver. The next class of probes I'd like to talk about are targeted probes. These are probes that are acting like an in vivo Western blot. Here, indium-111 DPPA labeled octreottate is utilized to check the expression levels of somatostatin receptor type 2. This was courtesy of Sam Machulifu's lab. In the top, one can see clearly that the probe is distributing throughout the body and then clearing through the bladder. When a tumor overexpresses SSTR2, the probe goes throughout the whole body, reaches the tumor, and is then internalized and trapped, while the probe is excreted from the rest of the body, giving you a nice clear image of the SSTR2 expression in the tumor by 72 hours post-injection. This data was confirmed with ex vivo analysis. If you don't want to work with radioactivity, you can switch radioactive labels for optical fluorescent labels. These allow rapid identification of target sites without all of the paperwork and potential danger of working with isotopes. Indeed, by looking at an X-ray image of a mouse that has a tumor on its neck and then injecting with fluorescently labeled octreottate, one can confirm that this tumor, as you can see here in X-ray, is high for somatostatin receptor type 2. One can also look at integrin expression through RGD-labeled peptides. These RGD peptides were labeled with a near-IR dye, injected into a mouse, and they were trying to track a known site of tumor cells. Alpha V beta 3 is known to occur at sites of neovascularization, as well as just being expressed on some tumor types. What was interesting about this experiment is that they saw fluorescence coming from an unexpected site, the upper paw. They then zoomed in and were able to discern that there is a nice hot spot here at the wrist. They could then guide histology by looking at the fluorescence image co-registered on the X-ray and confirm that there was hyperplasia at this site. Another group developed a chemical screen for peptides that should target alpha-4 beta-1 integrin. They then wanted to rapidly confirm that this peptide could target the integrin in vivo, so they biotinylated the peptide and then added streptavidin, which had been labeled with a near IR dot. As you can see here, they were able to clearly discern the tumor site from the surrounding tissue utilizing this methodology. In order to truly confirm that they had seen real targeting with this probe, they had to do several important negative controls. The first is to understand the distribution of their probe in a no-tumor setting and confirm there just isn't some other piece of anatomy here that is hot, such as a lymph node. Another important negative control for any in vivo experiment is to always acquire an autofluorescence image. Even in the IR, there's some autofluorescence, so it's important to empirically determine that noise floor every time you do an experiment. Another important negative control is just to inject dye alone, in this case, streptavidin dye. Tumors really take up things, as we've noted before, frankly, in an unpredictable manner. And so it's important to determine that your signal is higher than some non-specific uptake at the tumor site. In this case, it is. Another great biochemical negative control is to utilize a scrambled peptide or a D-amino acid peptide that shouldn't bind to the target site. And when they did this, there was really no targeting to a tumor. Another good biochemical control is that if a probe is binding to a low abundance receptor site, it should be blockable. And in fact, whenever they pre-incubated this tumor with a blocking antibody by injecting it into the animal, they were able to significantly diminish the amount of construct that targeted to the tumor. Another set of controls that they ran, which are also very good controls, are what I like to call genetic controls. Here, a tumor cell line that's known to be null for the target, in this case alpha-4, is put into the same animal, and then that tumor cell line has been modified such that it now overexpresses alpha-4. So in the same animal with the same dose of probe, you can clearly see that the alpha-4 is being specifically targeted versus just any run-of-the-mill tumor. The next class of probes that's very exciting are activatable probes. These are probes that are darker in their injected state, and then when an enzyme acts on them, they become bright. We've already seen one example of that in beta-galactosidase with DDAO galactose. In this example from the NCI Imaging Boot Camp, 4 T1 tumors were subcutaneously implanted in a nude mouse. These were known to exhibit MMP activity at their surface. 24 hours prior to imaging, MMP sense, a probe that works by having many fluorophores next to each other, thus decreasing their efficiency, was injected. If MMP activation is occurring at the site, those fluorophores will diffuse away from each other and become brighter. That bright dye is then eventually cleared to the bladder, and that's exactly what you can see from this in vivo experiment. The tumor is highlighted here, and the bladder, where the fluorophore is clearing from, is highlighted below. Almost everything that we've talked about so far has been looking at extracellular enzymes or receptors. Intracellular enzymes and receptors present a particular problem for in vivo work because your probe has to actually get across the cell membrane, target the site, and then clear away from the non-targeted sites. With activatable probes, you may not have to have them clear away because you want to trap that brightness inside the cell. In fact, this is how Bullock et al. Developed a nice probe for looking at caspase 3 and 7 activation. The first part of the probe is a permeation sequence based on a peptide variant that gets into many, but as we discussed earlier, not all cell types. There's a recognition sequence, in this case DEVD, that's clipped by caspase 3 and 7. Light then comes in during your experiments, excites the Fluor 647. That energy is then non-radiatively transferred to the QSY21 quencher, where it's then radiated away as heat rather than emission light. In the presence of active caspases, the fluorophore diffuses away from the quencher, is excited by 647 nanometer light, and then emits its normal 665 nanometer fluorescence, which can be detected as an optical signal. This was confirmed in vivo by looking at human colon xenografts on the backs of mice. Here, these human colon xenografts were infected by an amoeba, and you can clearly see activation in A and B. The biochemical control here is a D-amino acid DEBD sequence. If some other metabolic enzyme were causing the quencher and the fluorophore to no longer associate, it would show up in this. And so that's a really nice biochemical control for DEBDA specificity. Also, as I mentioned before, a saline-injected control to set the autofluorescence level is also key. The ability of this probe to detect activated at least cell death was confirmed by looking at tunnel staining from an independent histologist of these amoebic infections. In conclusion, having multiple imaging modalities allows for the capture of phenotypic as well as molecular information, such as looking at levels of receptors, enzyme activities with activatable probes, and promoters. By being able to capture and precisely co-register all of this information, you get the most complete understanding of your biological systems. I'd like to thank you very much for your attention, and you can feel free to contact me at my email address, seth.gammon@carestreamhealth.com for further information.